Good morning, children. Shubha teacher here. Here I am today with the third video of chapter Seed, Structure and Function. Till now, you have studied about the basic structure of seed and function of its various parts. Let us proceed with the conditions necessary for germination of seeds. There are three basic conditions which are required for germination: water, suitable temperature, and oxygen. Water is required for swelling and rupturing of seed coat. Now, before germination, seeds are soaked in water. When they are soaked in water, through micropyle, water enters inside it and volume of the contents inside the seed increase. That exerts pressure on the seed coat. As a result, the seed coat ruptures. That is, it breaks open. And that helps the growing embryo to move out of the seed. Next is, water is required for the action of enzyme on stored food. Now, we know that either endosperm or cotyledon stores food in the seed for the growing embryo. This stored food becomes easily available for the embryo by the action of enzyme, and enzyme acts on the stored food in soluble form. So therefore, water helps the seed in germination in a very great way. The next condition is suitable temperature. The optimum temperature required for the normal functioning of body of any living organism is 25 degrees centigrade to 35 degrees centigrade. This range is normal for all the living organisms, and in this condition, the cells work comfortably. The enzymes in their normal form, they help in carrying out various metabolic activities. Any temperature lower than 25 degrees centigrade or higher than 35 degrees centigrade becomes an uncomfortable condition for the living cells as well as enzymes. The next condition required for germination is oxygen. Now, we breathe in oxygen during respiration process, and that is used for oxidation of food. That is, food is broken down in the presence of oxygen to release energy, and with that energy, cells of the embryo grow, multiply in number, and helps in formation of a seedling. Now we go to the experiments on germination of seeds. There are 4 experiments which are discussed in your book. We will discuss each one of them in the videos. The first is to prove that water is necessary for germination. Second experiment is to prove that a suitable temperature is required for germination. Third, to prove that air or oxygen is necessary for germination. And the last experiment is the 3-bean experiment. We move to the first experiment to prove that water is necessary for germination. We know the 3 conditions which are required for germination: water, suitable temperature, and oxygen. Now, any of these conditions in an experiment, if it is missing, we then observe and prove that the conditions are required. Here the figure shows 2 beakers. Beaker A has got dry cotton wool at its bottom with some soaked seeds over it. The beaker's mouth is open, it means air is entering into the beaker and becomes available for the seeds. In beaker B, seeds are placed on wet cotton wool. So it means water is available. Again, the mouth of the beaker is open. It means air is also available. And both the containers are kept at normal temperature. It means conditions are normal. Now, this setup is kept for 1 or 2 days. After 2 days, we observe that the seeds in beaker A doesn't show any germination, whereas in beaker B, they have germinated. This happens because When we see the conditions available in beaker A, we see water is missing. The cotton is dry. Therefore, seeds do not get water here in beaker A. Whereas in beaker B, all the three conditions, that is water, cotton is wet, so water is available, temperature is normal, and oxygen is also available. So, because in beaker A water is missing, seeds did not germinate. Thus, this experiment proves that water is required for germination. Experiment 2 is to prove that a suitable temperature is necessary for germination. Now here in the figure you see there are 4 test tubes A, B, C and D. We ignore B and C here to understand better. Test tube A has got wet cotton wool and some soaked seeds over it. Mouth of the test tube is closed with air inside it. And this test tube is placed at room temperature. Test tube D has got wet cotton wool. Over that some soaked seeds are there. Mouth of the test tube is closed, so air is also inside it. But this test tube is placed inside a refrigerator. So it is under low temperature. Again, these 2 test tubes are placed for 2 days without disturbance. After 2 days, observation is done and it is seen that the seeds inside test tube A has started germinating, whereas seeds inside test tube D did not show germination. Now, the difference in the 2 setups is of temperature. Since seeds in test tube D were kept at a very low temperature than the normal temperature, therefore they did not show germination. Thus, this experiment proves that a suitable temperature is necessary for germination of seeds. With this, we come to the end of today's video. I have a small activity for you. The same 2 experiments which you understood right now, you have to do it yourself. 3 materials required are 2 glass bowls, cotton and some soaked seeds. These 3 materials you arrange at your home and read the 2 experiments in your book, listen to the video and understand it clear, follow the steps and perform these experiments at home. You will find it very exciting when you see the seeds germinating in various conditions. That's all for today, children. Thank you and stay safe.